The bulk of the cell membrane is made up of phospholipid molecules. These molecules are arranged in two rows. The molecules within the same row often exchange places with each other. They're constantly moving like a beaded curtain. The hydrophilic heads of the phospholipid molecule is facing outwards, and the hydrophobic tails, they face towards the interior of the phospholipid bilayer. The cholesterol that we see in green here stabilizes the phospholipid molecules so they don't move too much. Membrane protein is shown in blue here. There are many different kinds of membrane proteins. The next component is the carbohydrate chain. There are many carbohydrate chains all along the outer surface of the cell membrane. If a lipid is attached to a carbohydrate chain, we call it a glycolipid. When a protein is attached to a carbohydrate chain, we call the protein a glycoprotein. Now for the functions. Due to its hydrophobic interior, the phospholipid bilayer is very specific in what it can allow to pass through. Only very small polar molecules or hydrophilic molecules are allowed in. However, large polar molecules are not allowed to pass through the phospholipid bilayer. We call this property semi-permeability, and it also forms the bulk of the cell membrane. The next component is the membrane protein. There are many different kinds of membrane protein on a cell membrane. They could be a receptor, an enzyme, a channel, a gated channel, a cell adhesion molecule, or a cell identity marker. The next component is the carbohydrate chain. Carbohydrate chain, when attached to a protein or a lipid, they act as identity markers, cell-to-cell identification. And they also serve a protective function because there are so many of them on the surface of the cell membrane. Cholesterol's function is to stabilize the phospholipid bilayer when the temperature is high and the phospholipid molecules tend to move a little too much. What can and cannot pass through the phospholipid bilayer? Here is the phospholipid bilayer in pink. We need to remember two rules before we consider different kinds of molecules. The first rule is the phospholipid bilayer has a hydrophobic interior. The second rule is that the phospholipid molecules are constantly moving and exchanging places, which means very small molecules can make it through. So let's look at these one by one. Here we have oxygen, carbon dioxide, and water. These three molecules are very small, which means they can squeeze through the phospholipid molecules, the space between the phospholipid molecules. So they can pass through very easily. Now let's take a look at some hydrophobic molecules. Lipids are hydrophobic. They don't like water. Water and oil do not mix. So all lipids are hydrophobic. That would include steroids, such as hormones, and fats, which are triglycerides. All of these can easily pass through the phospholipid bilayer because they are hydrophobic. Let's look at ions. Ions are charged atoms, which means they are hydrophilic. Ions are also fairly big. They cannot fit in the spaces between the phospholipid molecules. So they don't satisfy both rules. As a result, ions cannot pass through the phospholipid bilayer. They are big and they are hydrophilic. The next example is glucose, which is a monomer. It cannot pass through the phospholipid bilayer because, like the ions, Glucose does not satisfy both rules. It is hydrophilic and it is too big. There are several different kinds of membrane proteins, so let's look at them one by one. The first protein on the extreme left is a receptor protein. You want to think of a receptor protein as a bouncer in a bar. Your bouncer checks your ID before he lets you in, so the receptor protein is like a bouncer. The second protein is an enzyme protein. The third is a channel. The channel is always open. The fourth protein seen here is a gated channel, which means the channel can open or close depending upon the stimulation. The fifth protein is a cell identity marker with a carbohydrate chain attached to it. This allows cells that are similar to identify each other, and then the last protein, cell adhesion molecule, helps these like cells to stick together. We call this CAM, cell adhesion molecule.